So today on the three major aphid pests with small grains in the U.S., be the Russian pig aphid, greenbug, and the bird cherry-oat aphid, and also a new invasive aphid pest, the hedgehog green aphid. Thank you.
Like on the, it's like on the PowerPoint, it's like on the PowerPoint.
Little person, I don't know how to do it.
That didn't.
It didn't.
Down. Down.
Okay, down works. Okay, sorry. Anyway, I wanted to start by making sure that I acknowledge all of my field cooperators. Most of what I'm going to speak about today would not have been possible without their assistance, especially because I'm from Oklahoma. I did a lot of work with spring barley, but we don't grow spring barley in Oklahoma. So I had to be in a lot of other different states to do a lot of my field research. You can see there's quite a few states involved. In the beginning for the springs, mostly worked with people in Colorado, Wyoming, also Idaho and Nebraska. And as I transitioned into some of the winter barley work, it included all of the states that you see. A lot of these people are actually entomologists, a good mix of entomologists and breeders. I also had excellent mapping cooperators who helped me when I needed to identify genes and map those genes. ARS in Fargo and ARS in Stillwater did all the mapping. Okay, so we'll start with Russian wheat aphid. Just a very brief history. It was an invasive pest of small grains, first reported in Texas in 1986. It was found in all states west of the 100th meridian and 3 western provinces of Canada by 1988. One of the reasons that it spread so quickly through that area is because there had been a federal program to plant CRP grasses, and they were very hospitable to the Russian wheat aphid. And from 1986 to 1991, combined direct and indirect losses were in excess of $1 billion in U.S. small grains, and all of U.S. barley cultivars were susceptible to Russian wheat aphid. So it was really a big problem then, something we were very concerned with. So greenhouse seedling screening of the entire NSGC of Mornhinweg was done by entomologists at my lab in Stillwater, Dr. Jim Webster. And over time, we identified a total of 116 accessions with some level of resistance. Only about a third of those had high resistance. So we also did some field tests to show that greenhouse seedlings accurately predicted field resistance, which is great. That meant we could just go ahead and proceed with all of our screening and be confident that our resistant lines were going to do well. So then homozygous resistant and unadapted germplasm lines were selected from each of these accessions, and resistance was transferred into adaptive barleys. Can't read my last line. So anyway, eventually we released germplasm in cultivars as well. Then in 2003, a new biotype of Russian wheat aphid was found that overcame the newly deployed single gene resistance in wheat. So something people didn't think was going to happen because it was an introduction, didn't expect this change to happen so quickly. And by 2008, there were a total of 8 Russian wheat aphid biotypes that had been identified. So the genetic diversity for resistance was essential for protection from crop loss. Now, the barley lines that I first began to work with had multiple gene resistance, and as yet they have stood up to all the U.S. biotypes that we against. Resistance to some also have resistance to some of the predominant world biotypes, which would be Iran, Mexico, Chile, and I don't remember the fourth one. Anyway, that is was very encouraging. So the damage caused by the Russian wheat aphid, they remove sunlight from the vascular tissue. They also inject saliva, which induces senescence that allows them to take advantage of increased translocation of proteins. And the resulting damage is leaf chlorosis, streaking, and rolling, in which the new leaves do not emerge or unroll upon emergence. The rolled leaves are essentially a pseudogall, protects the aphids from natural predators, wind and rain, and contact insecticides. So in the beginning, when we didn't have resistance, we had to use contact insecticides. They have to have several applications. So if we switched to systemics, there was a lot of concern of where those ended up in the grain. So it wasn't something that we wanted to do. The greatest yield reduction is from the trapping of the heads and results in deformed heads and severely reduced fertility. Highly resistant lines have little or no chlorosis or streaking, and they have flat roots, resulting in little or no real reduction. So the first two lines that germplasm lines we released were unadapted spring barleys, stars 901B in 1993 and stars 9577B in 1995. The stars actually stands for Stillwater-ARS. It's kind of a crazy name. But anyway, and the first two numbers are the year of release. So these are unadapted barleys, and we were, the 901B was just a little bit more resistant than 9577B. We didn't know at the time what the genetics involved were, but over time we did discover that they both had multiple gene resistance, RDN1, RDN2, and RDN3 in 901B, and just RDN1 and RDN3 in 9577B. So there really was a little bit of a difference in the resistance, explained genetically. But they're both still highly resistant. And also these are unadapted barleys, and they were not very agronomically pleasing. So a lot of the breeders were really leery about using them. So we decided to do some pre-breeding for resistance in some adapted lines, and had a lot of the mostly AMBA-associated breeders send me their elite lines so that I could transfer genes into those adapted germplasm. So as a result of that breeding program, we had 19 6-row spring barley germplasm lines. They involved 19 sources of resistance in 4 multi barley backgrounds, more XXL, standard, and robust, which were at the time elite lines. And 2 of those 19 actually had resistance from B 9577B. So we really didn't know what resistance we had in the other 17 sources, but we were shooting for genetic diversity and just hoping it out. Surely some other genes involved there. They also released 17 2-row spring multi-barley, and they have 17 sources of resistance in B1202, cress, crystal, and 2 of these lines also have resistance from sensitive. Then we also worked with feed barleys and released in 2005 7 6-row winter feed barleys, and they had 7 sources of resistance. And again, we weren't sure that we actually had different resistance genes in these lines, but we were hoping for the best. At the time we were doing this, mapping wasn't that big, and so it really wasn't something that we could take the time to discover first before we transferred the resistance. So we just kept moving forward and just keeping our fingers crossed that we were going to have genetic diversity. Okay, we also released 7 2-row spring feed barleys, and they had 5 sources of Russian weighted resistance in 3 different feed barley backgrounds, Bowman, Hector, and Otis, and 2 of those 7 actually had the resistance from the 77. Okay, so a total of 50 germplasm lines were released between 2005 and 2006. And again, the genes were involved in resistance and yet to be identified. However, we did a separate test, subset of 46. It was a association mapping project, and we found multiple loci on 6 of the 7 chromosomes of barley. So that definitely indicates we had genetic diversity. The first RDN1, RDN2, and RDN3 were on 3 separate chromosomes, but we found associated genes with resistance on other chromosomes too. So we're feeling pretty good that we do have some genetic diversity out there. Okay. Then also we did find that we did have genetic diversity for some of the rural predominant biotypes, which is very encouraging. And I think about 17 of those lines actually have resistance to all 4 of the foreign biotypes, which is very good news. Okay, switch to greenbug. Brief history. Greenbugs have been in the U.S. since 1882. And the first biotype was identified in 1958, and they gave it the designation of B, just considering that everything before it was an A. And from 1961 to 1997, 10 more biotypes were identified, 13 more in 2010, and another 6 more by 2016. So you get the picture that this biotype situation is pretty serious. All right. It was something that we had to definitely strive for genetic diversity. It was proposed that diversity among greenbug clones cultivated native grasses is an indication that they act as a reservoir for biotypic diversity. Okay, so greenbug infestations can occur in central, northwestern, and southeastern U.S. Most often the problem would be in the southern plains and a little bit in the Pacific Northwest. In outbreak years, it can extend all the way into southern Canada. And outbreak years are usually occur only at summers. It gets followed by, I can't remember what the last is. But anyway, it hasn't been a problem much lately. And in Oklahoma, the growers just don't mind spraying for it. But if we're going to be working with, say, a malting barley, we certainly don't want to have to be spraying. So we're still trying to find natural control of aphids. Okay, greenbug feeding causes seedling death or decreased yield, depends on the timing and intensity of the infestation. And they inject a saliva, which produces, induces senescence. So that's not just straight assimilate uptake, but it's because it's causing senescence that we have the damage. Yield losses of 35 to 65% or 6% have been reported. We have two genes have been identified, RSG1 and RSG2, and all of the current greenbug resistant varieties carry the RSG1 gene. Okay, so we had RSG1 resistance identified in 1945, RSG2 resistance in 1984. All greenbug resistant cultivars again carry the RSG1 gene. So recently we've discovered new greenbug resistant genes. 3 of them are allelic to the RSG1 and RSG2, but we discovered a new RSG3 gene, and we think we have a new RSG4 as well. We're just not finished with all of our testing for that. So 2 of those allelic genes were actually identified in the Fall Barley Diversity Collection reporting spontaneum that Brian's going to talk about. Okay, as far as greenbug germplasm lines, because we already had RSG1 in cultivars, we decided to concentrate on RSG2. RSG2 came from PI 426756, just segregating for resistance. So we decided we would try to get that in a stable condition, and we did that and released it as a spring 6-row spring line. And then we also released it as a 6-row winter line by West. So this may not seem like such a big deal, but I can tell you that this fall, when I was screening all my lines, which I do thousands, the entomologist accidentally gave me the wrong biotype. And it was one of the new ones, Texas 1. And it wiped out everything, except the few that had RSG2. So this can happen in the field. It's a real thing, and I'm really glad that we have these lines out there for people to use for breeding groups. Okay, so we also had some lines that we developed that were Russian-related and greenbug resistant. So there were 8 6-rowed winter feed barleys that had both resistance. 8 that involved 8 resistant sources of Russian and the RSG1 resistance. We also had 8 hollows or naked barleys that were released that resistant to aphids and 5 resistant sources and RSG1 resistance. So while we were developing all the pre-breeding germplasm lines, we came across some that were really good performers. We decided that we could release them as varieties. So we did that. There were 4 spring 2-row feed barleys that were resistant to Russian aphid: Burton, RWA 1758, Sydney, and Stone. And you can see that all 4 of those have resistance from either 577B or 93. So, and you may not know the importance of Otis because that's a kind of an obscure variety from Colorado, but it's highly, highly drought hardy. And it had a significant role in the rotations in the high plains of eastern Colorado, western Nebraska. Russian wave wiped it out. So the growers actually came to me asked them, please do something about it. So we did release those 2 varieties, probably not widely grown, but they're grown in that area. And then we also released a winter 6-row feed barley mesa that has a different source of Russian weed resistance in the Schuyler background. And that was developed in Western Colorado and is currently being sold in Colorado. So then we came to, this is post-90 was actually released by us, but it was a long time ago. That is the one that has the RSG1 resistance. And they're actually the most important one. You can't see it on there. Is USDA-ARS, which is resistant to both Russian weed aphid and greenbug. And it is a winter feed barley. Okay. Oh, there we go. Okay. So Mesa had a different source of resistance to 9301Z, and so does USDA-ARS, even another source of resistance. So pretty excited. No, it's not advanced. No, it's not working. Okay, all right, so let's switch to the vertebrate aphids. Do a brief history on that. It was first found in North America in 1855. It's been around a long time. It's a dual threat to barley, not only through direct feeding, but also as a very efficient yield losses from combined effect of BCOA and BYDB and winter barley can be as high as 8%. Long contact with the infected plant is required for the aphid to actually acquire the virus. The time necessary for that acquisition period can vary from 15 minutes to 1 to 2 hours of feeding. And temperature and age and infection stage of the source tissue can also affect acquisition and transmission. There's a lot of factors involved in this. Reducing the ability or desire of the aphid to feed on the plant for extended periods would reduce the level of virus in the aphid. Probability of the aphid for transmitting virus. So we're not looking for tolerance resistance as we have in all the other aphids, because that would just transmit the virus. So we're going to have to look for something different. Okay, BCOA feeding damage is asymptomatic, so it does not inject a toxic saliva. And less growth of barley due to feeding is caused by loss of symptom. So it is different than the other aphids. Feeding also inhibits root growth that can decrease crop tolerance to abiotic stresses like winter kill. That's a serious problem if you have an infestation involved on the barley. Yield losses from BCOA feeding alone has been reported 15 to 50%, but that can vary from field to field and year to year. Okay, so established aphids seedling screening protocols that rely on visual symptoms are not as effective for BCOA. I've been working with this for years, trying to come up with something that does work. Okay, so reporting, when you see in the literature reported resistance, it's most often measured as antibiosis, which is the negative effect of the plant defense on the aphid biology. It's incredibly labor intensive. There's no way you can mass screen. Right? Take forever. Besides that, this is a term I like to use. I'm not sure if it's a meaningful resistance that will actually protect against yield losses in the field. We have no idea. So I think I would have nothing to report here. But I have actually gone ahead and screened. I've screened up to 12,000 barleys from the National Small Grains Collection, and I'm doing it visually, but I'm not rating them to scale. I'm simply looking for ones that survive and or ones that appear to be non-preferred. So I have worked with one line for years, and I call it BCO-R001. And we have done studies to show that in the greens that it protects against yield loss. And it's also shown resistant to compared to a susceptible check based on morphological and zoological traits. And the aphids were less than 50 plant, but it did not tolerate the high rating numbers. So it's indicative of perhaps non-preference, and whether that's going to be good enough. I'm not confident about this line like I am about other things I work with. It also isn't stable. I can have a resistant plant that I save out of screening. It's progeny or not resistant. Some of them will be, but not all of them. So it's very complicated. But I'm excited to say that I have recently found a line that you can visually see is non-preferred, and we're progressing with that, trying to release that would actually be what we need for bird cherry. Okay, probably already said this, but anyway, tolerance resistance would not be preferred as it would increase transmission of the virus. And cyanosis for non-preference could prevent or reduce the feeding and thus the disease transmission. And pyramiding of anazinosis and anabiosis with disease resistance would offer the best protection. In the literature, it looks like anybody that works with the disease is also saying this is so complicated, we're going to have to rely on aphid resistance. And I'm saying aphid resistance is so complicated, we're going to have to rely on disease resistance. So somewhere in the middle, we've got to all decide together and pyramid those genes. All right, switching to the new aphid, the hedgehog brain aphid. It is a serial pest worldwide and also a dual threat through feeding damage and as a vector. It was discovered in South America in 2002 and it spread throughout Argentina's cereal producing regions by 2006, infesting wheat, barley, and bog grasses, causing extensive damage. HGA has 52 hosts worldwide and has been found in 14 hosts in the US, including Barley. Okay, it was first detected in the US in 2007 in California, and has since been found in Colorado, Utah, New Mexico, and Wyoming, and is well adapted to the high plains and Rocky Mountains. It's kind of the same area that the is adapted. But a recent survey found it to be established in limited colonial populations on hog grasses. Seems to be becoming smaller and smaller. It's area of adaptation, but I'm convinced that we've actually surveyed everything that we need to survey. So we're still working on that. At present, it's not considered a threat to cereal production. Okay, so HGA also ingests phytotoxic saliva, same way as the other greenbug and Russian wheat aphid, and resulting in chlorotic spotting. Yield loss is attributed to photosynthetic area, resulting in poor growth, and in the late season infestation, reduced green. HGA is a vector of volatile and can cause injury. Gene restrictions are currently prohibiting any mass screening, at least my meeting in Oklahoma. But we did do a very teeny tiny experiment where we took some of the lines that had the major aphid resistance genes for other aphids, RDN1, RDN2, RDN3, and RSG1 and RSG2. And we screened inside in a quarantine approved room in a really small study. But I'm happy to say we actually found some resistance. So at least it's in the seedling stage. So Skylar and Marks are susceptible. Stars 93OB, 957 and Mesa all have Russian aphid resistance genes. Stars 1501B is RSG2 and it's not resistant. So the two lines that actually have RSG1 resistance is 90, and the far one there is actually USDA Fortress, actually showed some resistance. Oh, there's much more that needs to be done on this. I'm not able to do it still. Okay, summary of vulnerability to aphids. Genetic diversity exists in available Russian aphid resistant and greenbug resistant germs. Genetic diversity is very limited for resistance to BCOA and HGA. Introduction of new aphid pests or different worldwide types of aphid pests is always a possibility. And more virulent biotypes can arise through interbreeding sexual forms from isophenol common threat to the deployment system. Aphids are predicted to become more abundant with warmer temperatures due to climate change, and are expected to have changes in persistence, abundance, dispersion, and migration. And some good news. Opportunity for biotype shifts that are fueled by environmental factors where one biotype survival is preferred over another also can increase change. Genetic diversity for resistance is essential for this thing. Production. Okay. So my 5-minute warning. Slide. Okay, so future research. Going to continue to search for BCOA resistance. Up at the corner there, you can see a picture which I should have shown earlier of the non-preferred new line. You can see it there or not, but definitely are not aphids on that one line. That's encouraging. Transfer of BCOA resistance into adapted lines. I'm already working on that. I've already crossed this line and our line into some of the more actually into some spring pressure resistant germplasm. Release of American barley germplasm with greenbug resistance from genes. We'll have working on that. Field testing and release of greenbug resistant from Walton barley for 7 plants. I love this one. I'm working on that down there. Some lines divided. Right, and continue surveying for HDA and determination of damage potential on cultivated barley. Just want to give a big thank you to Amber. There are quite a few lean years that my program would not have survived without the help of them. I'm so grateful for that. And also now that we have the new Barley Pest Initiative, that funding is short of my position, and now they are saying they will replace me when I retire. So that makes it easier for me to step away. My unfinished research on the table. Anyway, thank you very much.
Time for some questions.